Chapter 2, Prokaryotic Structure. All living things are composed of living cells. Cells are the smallest units that perform all vital physiological functions and maintain the properties of life. The properties of life are metabolism, reproduction, differentiation, evolution. Metabolism can be defined as the ability of organisms to take in nutrients to provide energy needed to grow, reproduce, and be responsive. Be responsive means respond to the environment. This generally involves the release or use of chemical energy. Non-living things do not display metabolism. Reproduction means increase in number, producing more organisms themselves. Reproduction may be accomplished asexually, alone, and sexually, with a mate, to allow cells to produce copies of itself. Differentiation is the process of transforming an unspecialized cell into a specialized cell. Populations of living organisms have the ability to adapt to their environment through the process of evolution. There are two types of cells: prokaryotic cells, which consist of archaea and bacteria; eukaryotic cells, which consist of plants, algae, fungi, protists, and animals, a variety of animals. As discussed in Chapter 1, we compared prokaryotes to eukaryotes. We said that prokaryote comes from the Greek word for pre-nucleus, before the nucleus, and eukaryote comes from the Greek word for true nucleus. Continue to compare prokaryote versus a eukaryote. A prokaryote, again, is before the nucleus, so it has no defined nucleus. The DNA is circular. Our DNA is linear, so prokaryotic DNA is circular. There are no histones. There are no membrane-bound organelles. The bacteria cell wall is made up of peptidoglycan. The archaea cell wall is made up of pseudomurin. They divide by binary fission, which means that they divide into two cells during cell division, and they are smaller than eukaryotes. Eukaryotes is true nucleus are true cells that have a nucleus with a nuclear membrane and membrane-bound organelles. Membrane-bound organelles are specialized structures that act as tiny organs to carry on the various functions of the cell. So they also have histones. The cell wall is made up of polysaccharide. They divide by mitosis or meiosis. So we've talked about mitosis back in Biology 1151 or Anatomy and Physiology 1. And they're usually larger than prokaryotes. In this chapter, our main focus will be focusing on prokaryotic cells. The shapes of prokaryotic cells and size. The average size is about 0.2 to 1.0 micrometers to about 2 to the 8 micrometers. Most bacteria are monomorphic. Monomorphic means that they maintain a single shape. A few are pleomorphic, meaning that they have many shapes. Morphology means shape. We already talked about coccus in Chapter 1. What does it mean when we say coccus? If you said spherical or round, you are correct. So here is a picture showing you a cocci cell, how the shapes are circular. Bacillus is rod-shaped. So here you can see you have the nice rod-shaped. And spiral are curves. There are some less common shapes or unusual shapes. You have the star-shaped cells that are found in the genus Stella, and then you have rectangular flat cells or square cells found in halophilic archaea called haloarcula. So here's a picture of that. So now that we understand the shapes, coccus, bacillus, and spiral, let's take a look at the arrangement of these shapes. They can be arranged in pairs. Pairs are two. Okay, so diplococci, so you see two cocci here, and diplobacilli, we have two bacillus here. They could be arranged in clusters, and we said that the word for clusters was staph, so staphylococci. So staphylococci looks like a cluster of grapes. There's no staphylobacillus. There's only staphylococci. Chains would be strep. So we look at strepto. So streptococci is arranged in a chain fascia. In round circles are arranged in a chain fashion, as well as streptobacillus. The rod shapes are arranged in a chain fashion as well. Looking at the arrangements for spiral, which is curves, you can have spirillum, vibrio, or spirochete. Vibrio is when it has less than one term. So vibrio is less than one term. Spirilia or spirillum is helical, is fairly rigid, and usually movement is provided by a flagella. And then you have spirochetes, which are also helical and flexible, but movement is provided by an axial filament. We are going to now take a look at the structure of a prokaryotic cell. We're going to start with the external structures first, and then work our way into the inside. Glycocalyx. The glycocalyx is a sugar coat or gelatinous sticky substance that surrounds the outside of the cell. There are two basic types, the slime layer and the capsule. The slime layer is loosely attached to the cell and not well organized. The capsule is closely attached to the cell and well organized. Both the capsule and the slime layer are made internally and secreted to the outside of the cell. The general function of the glycocalyx is the same for both the capsule and the slime layer. Adherence, attachment. Extracellular polysaccharide allows the cells to attach. Streptococcus mutans is responsible for tooth decay. The capsule anchors the bacteria to the surface of the tooth where they produce acid and cause decay. If it weren't for the capsule, S mutants would be dislodged by the saliva and we would suffer much less tooth decay. Capsules prevent phagocytosis. It's a virulence factor. Virulence factor means that it protects the bacteria. So capsules make the bacteria anti-phagocytic. So it provides a protection of the pathogen from the host immune system. Since the chemicals are similar in many capsules, are similar to those normally found in the body, they prevent the bacteria from being recognized and devoured by the defensive cells of the host. Unencapsulated strains of these bacteria do not cause disease because the body's defensive cells will destroy them. So if they lack the capsule, they will not be able to hide from the immune system. Examples of bacteria that have these capsules is Bacillus anthracis. This causes anthrax. Klebsiella pneumoniae. This causes pneumonia. The last function is protection from dehydration. It has the ability to absorb moisture, which is protective against drying. Flagella. A flagella is located outside the cell wall and is a long filament that propels some prokaryotes. A cell's motility may enable it to flee from a harmful environment or move towards a favorable environment where food or light is available. The function of the flagella is movement. It acts as a propeller and helps to move the cell. The structure of the flagella consists of three parts. The filament. The filament is the whip-like shaft that extends into the cell's environment. It's made out of flagellin protein chains. The filament is attached to the hook. The hook is a protein structure at the base of the filament. The protein hook is then anchored to the wall and the membrane by the basal body. The basal body holds or anchors the flagellum to the cell wall and the cell membrane. So here we'll see we have our filament, and then we'll see that we have the hook, and then we have our basal body. There are 4 different flagella arrangements. The first one is monotrichous. Monotrichous means one flagella, so you'll see here this has one flagella. Amphitrichous means that you have flagella at both ends. Peritrichous means that you have flagella all around. Lophotrichous means that you have 2 or more flagella at one end of the cell. So there's the lophotrichous. The last one is called atrichous, and atrichous means that it lacks flagella. Multicells. The flagella is going to rotate clockwise or counterclockwise to run or tumble. It's going to move towards or away from a stimuli. This is called taxis. Taxis is the movement towards or away from a favorable or unfavorable environment, respectively. Both runs and tumbles occur in response to stimuli. The stimuli is the signal that stimulates movement. The stimuli can act as an attractant, which is positive, or repellent, which is negative. There are 2 types of stimuli. Chemical leads to chemotaxis. Bacteria sense certain chemicals and swim towards regions that contain more nutrients and away from regions with toxic materials. Light leads to phototaxis. Photosynthetic bacteria swim to regions of optimal light intensity and quality. Understanding runs and tumbles. The types of movements that a bacterium can have are a run. A run is a rotation in one direction resulting in movement of the cell in a single direction for some time. So here you'll see that we have a run. So it's moving in a single direction, sort of like in a straight line, and this is also a run. A tumble occurs when rotation switches directions, resulting in abrupt random changes in direction. So here we have a run, and now there's going to be a change in the direction of the way that the flagella is going to turn, and that's going to cause a change in the direction, leading to a tumble. Then we have a run, then up, the flagella is going to change direction and turning, and we're going to see a run. The runs and tumbles are random without stimuli. The ratio of runs to tumbles increase when stimuli is present. So, if the stimuli is an attractant, there is more runs and less tumbles. If the stimuli is a repellent, there is less runs and more tumbles. So where are the receptors that respond to the stimuli? The receptors for the stimuli is in the plasma membrane of the cells. Active filaments are also called endoflagella. And this is what we see in spirochetes. They move the spirochetes, and there are two bacteria that have axofilaments. Treponema pallidum causes syphilis, and Borrelia burgdorferi causes Lyme disease. Axofilaments are fiber bundles beneath a sheet, anchored at one end of a cell. Rotation of the filaments leads to a spiral movement of the bacteria. So rotation causes the cell to move. So these axofilaments will be rotating, and then that causes this bacterium to move. Other external structures. Most gram-negative bacteria have hair-like appendages that are used for attachment rather than for motility. These hair-like appendages consist of a pilin protein arranged helically around the cell. They're divided into two types. The first type is called fimbriae. Fimbriae is found at the ends of the cell or distributed evenly over the entire surface of the cell. It is used for adherence to the mucous membranes or surface. So it allows for the bacteria to be able to attach or adhere to surfaces due to those sticky Bristol-like projections. And you can see a picture showing you that fimbriae. Neisseria gonorrhea, which causes gonorrhea, have fimbriae attached to them so they can move through or colonize mucous membranes. So Neisseria is able to colonize the mucous membranes of the reproductive tract by attaching with fimbriae. Pathogens must be able to adhere to their host if they are to survive and cause disease. Neisseria cells that lack fimbriae are non-pathogenic. So if they didn't have their fimbriae, they would not be able to attach and cause gonorrhea. Pili is longer than fimbriae. It's used for attachment to other cells so there could be a transferring of DNA between cells. So pili is a much straight hair-like appendage, okay, and it allows for DNA to be transferred from one cell to another. So a perfect example of this is a sex pili during conjugation. So here's a picture showing you that straight pilin protein going all the way across. And there's a YouTube video here that you can watch to see how conjugation actually works. Here ends part A of chapter 2.